Jnanam Paramam Dheyam. Knowledge is supreme.
Welcome to MOOC course on applications of interactomics using genomics and proteomics technologies. In the previous lecture, we discussed about SPR technology, especially Bayer core platform to perform surface plasmon resonance based experiment. Dr. Uma Sinha Datta, an application scientist and a trainer from G Healthcare, gave you the basic idea for doing these experiments. Today she is going to continue where it was stopped in the last lecture, and she is also going to provide you a hands-on session on Bayer core technology. So, let us have Dr. Uma Sinha Datta for today's lecture.
So, I pointed out one thing to you: when you are directly immobilizing your ligand, it is covalently linked, right? And once you immobilize it, it is immobilized for you good. You cannot use the chip to immobilize anything else, right? You use the same ligand to do your experiments in that. But there is a smart way to actually save your or use your chips more judiciously, you know. So, when you are actually immobilizing it and you are playing with the RUs, how much to immobilize, and you do not know how much you would like to immobilize, or you are attending some part of it. Say we decided to immobilize 600 RUs, but how would you go and immobilize if you, then you have to actually look at your contact time, how do you attend that 600 RUs, right. So, there is a wizard that we have, which we call it as this control wizard, and when you set up your immobilization, you can say aim for immobilization, okay, rather than putting a contact time for immobilization. And when you do so, what happens is your system actually pulses small volumes, say around a microliter or so, on the surface and does a pre-concentration for you. And with the rise in the RU, it calculates whatever you have aimed to attain, around 600 RUs. It will calculate and let you know in the first 6 or 5 to 6 pulses, it will let you know whether it is attainable or not. If it thinks that it is going to attain, then it proceeds to the EDC NHS step. Before that, it does a quick NaOH wash to remove all the electrostatically bound ligands, and then you do an EDC NHS, and then you pulse your ligand based on the information that you had collected, and then finally you do the ethanolamine step. In case it decides that the concentration that you have supplied is not enough to attain your 600 RUs, whether it is more or high, it is going to abort your cycle post here. So, your chip is actually intact in this case, okay. And this is a very useful tool to use when we are because the chips come really expensive. There is another one which is not added here, which is the CM7, typically used for the small molecules. Okay. A little bit more detail on the chips. So, you know the basic structure of the chip. The chips are actually on the glass surface on which there is a gold coating, then there is a linker molecule where the dextran molecules are stuck to it, and the dextran molecules have carboxymethyl groups, and in these carboxymethyl groups is where your covalent linking happens. Okay. The CM5 chip is actually our most versatile chip. Most of your even now when I do my first experiment in Bayer core with a particular system, I would straight away go with CM5. If it does not work, I then go and choose a different chip, because it's been proved to be very, very versatile to work for most proteins, okay. And it can support almost all kinds of covalent chemistry there. Now, a slight variation of the CM5 is actually CM3 and CM4. Okay. So, they are exactly the same. It is just that the length, in one case the length of the dextran is the same as CM5, but the carboxymethylation, the number of degree of carboxylation out here is less than in CM5, whereas in CM3 the degree of carboxylation is the same, but the length of the dextran is actually smaller. Okay. So, this is used mostly to, if you want to have work in a low Rmax region, if you have a CM5, sometimes it's sometimes difficult to attend low Rmax, whereas in this case it's much easier to attend the low Rmax. The smaller size of dextran here helps to do your assays in larger molecules, like for example you are working with viruses or cells where the size of your cell is quite big, and it moves away from the surface. So, the surface plasmon resonance will happen quite far away from the surface, and where your the SPR phenomena reduces as you go far away from the surface. So, that is where you actually use a smaller branch, so that you can keep your proteins or your cells closer to your surface. C1, I think we discussed about that, which is the dextran is all gone, it is only the carboxymethyl groups which are stuck on the surface. In cases where your dextran is causing non-specificity, you can use this kind of chip. Right. This is your streptavidin. This is streptavidin chip for your bioethylated protein. But remember, this protein, the streptavidin biotin binding is extremely high KD. I think it's in the range of 10 to the power minus 12 or so, which is actually very, very close to being a covalent linkage. So, once a streptavidin and biotin links, you can never open it up. So, you have to be a little careful when you are using this chip, because once bound it is good to go, like a covalent chemistry. NTA, this is for the histidine tag. This is reusable, you can recharge your chip all the time with the regular chemicals that are available for NTA columns. HPA, this is for hydrophobic interactions. You can coat your hydrophobic proteins on here. It's a flat hydrophobic thing. This is to make in this case you can only make a monolayer of hydrophobic layer, whereas here you can make a lipid bilayer for using the liposomes. If you have the liposome mixture, you can make a lipid bilayer kind of a structure on this thing, and then have the protein studies done on them. So, that is the summary of all the chips. I am not going to go through this anymore, because we have talked about the various kinds of chips now. Unless you have any questions, I can answer them. So, that was all about the surface preparation, which one should you use, what is your ligand, the PIs that is related, important, how you can optimally use your chips by not wasting them, right. And we also learnt about the Rmax, in which Rmax you are going to work, things like that. So, the next one is sample injection. This is rather a simple thing, it is just that you are passing your analyte over the ligand that you have prepared. Now, if you remember, we talked about that SPR is actually a refractive index measure, right. The change in mass causes a refractive index change, which you are reading, and so difference in concentration in your buffer could also actually change your response unit that you are reading, right. So, what we call it is actually a bulk effect, when you are passing your analyte is in a particular buffer, the buffer also gives you some response unit. Okay. So, if you look at this graph, the blue one is just the buffer running without anything. Okay. So, it has a teeny bit of response unit. The red one is actually your binding as well as your bulk. So, you are looking at a response unit which you have received after binding, as well as you are looking at the bulk. If you subtract these two, you actually get your true binding. So, all SPR experiments should have a 0 concentration which is subtracted, and the buffer needs to be exactly the same. So, whatever is the running buffer or the 0 buffer has to be used to make the sample, right. And then you use a reference surface. This is to remove non-specificity of your analyte with the surface, right. When you are passing your analyte over the ligand, somebody asked me, I think from within you, that how do you make sure that it is binding to the ligand and not to the surface, right. So, you have another blank surface which is just before your active surface, and there you actually see if there is any non-specific binding that is happening with your dextran, and you subtract that, which we call it as a reference surface. So, when your analyte passes to the reference surface, you see a certain pattern of response unit. This is your active surface, and when you subtract this is actually your the real binding minus the non-specific binding. In some cases, if you have too much non-specificity, it might interfere with your binding. In those cases, you are supposed to use other additives like detergents, or there is something called an NSB reducer, which is nothing but free carboxymethyl groups which are mixed with your analyte to reduce the non-specificity. Okay. There are 3 different ways to produce or make your reference surface. There are various ways you can have a reference surface. The first one is unmodified. You do not have to do anything. Okay. In most cases that works, but if you are not happy with that unmodified, you can also treat it exactly how you immobilize. So, you activate and deactivate without passing the ligand. So, you are treating the surface exactly the same way that you have treated your active surface. Okay. So, the first one is unmodified, the other one is activated and deactivated. And the third variety can be, you can have a dummy ligand, a knockout ligand which is knocked out of binding, that is also possible. Okay. So, if you have it, if you have the luxury to have that, you can also have that, another way of doing the surface. Certain sample considerations, not I mean they are very critical, but also very logical. I mean the sample has to be homogeneous, you cannot have particulate matter, or they will create spikes, dirty sensorograms, eventually difficult to evaluate, right. The quantity of analyte also has to be extremely, it is very important, particularly in certain applications like kinetics. The concentration needs to be determined very accurately. Wrong concentrations will land up giving you, different values. Okay. You need to make sure your analytes are active, they are free of aggregates. If it is aggregated, then you get higher response than usual, which is not right. Okay. And buffers, that is again what buffers to use, that is also very, very important, because sometimes your interactants may require certain metals for binding to happen. That is something that you need to find out from literatures, whether you want to add some additives to promote your binding. So, these are some things that need to be considered. And also injection time, we spoke about how much of time does it take to run a full cycle, right. And it is a very generalized question, because it depends on your association and the dissociation that we understood, right. So, if it is a very quick associator and a dissociator, you would obviously need a smaller time, and if you have a very slow association and dissociation, obviously the time needs to be increased. Buffer requirements, the buffers that go in should be 0.22 micron filter. They are of course available to buy from GE, but it is not a big deal. I mean, you can make it yourself as long as your chemicals that you are buying are of good quality, and just pass them through 0.2 micron. Do make sure you have P20, which is a detergent and it is a non-ionic detergent, because you do not want any ionic detergent running over the surface that can spoil your things. It is important to have a detergent to remove the stickiness, because the proteins are quite sticky and they stick to your flow cell, and for the maintenance of your system it is important also. In some cases where you do not see binding, you might want to remove the P20 and do your experiments and see if it is working, but make sure after that you run through a good maintenance cycle. Okay.
What is P20?
P20 is a detergent. It is a non-ionic detergent. Sorry, I don't have the full name. So, once you have your immobilization ready, you pass your sample exactly that the experiment that you did today in the lab. You know, you did a 0 concentration, you did a low concentration, and you did a high concentration. You need to test the surface exactly that way, because the first thing is you don't want to use huge amount of analyte, which could be very expensive for you to get it actually to set up so much of run, and then you realize your ligand was not proper or your surface was not right. So, you test the surface once before you set up a big kind of experiment. So, you run 2 different concentrations, low and a high. First, you get to see the shape of the curve, which gives you a lot of information, like we discussed. You know, the shape of the curve you get to know, and you get to see that exactly what you were expecting, you have got that. The other thing is you see a dose dependency, right, that a low concentration, higher concentration, you see a dose dependency. The third point is you have also calculated your R max, and based on that you have had immobilized your ligand, and with that are you getting your R max, are you getting your RU or not. So, these are very, very important things to keep in mind when you are setting up your assays, because if you don't, if something is actually not falling in place, it's time to check right there rather than setting up the whole thing and going. Okay. So, that was all about the sample injection. So, the last one is your regeneration, right. After your association, dissociation, before you go to the second cycle, you actually find out the regeneration, which is stripping, removing all your analyte from the bound ligand. Okay. I think you saw this, it is just removing all your bound analyte from the ligand. So, there is a way to check whether your regeneration conditions are fine or not. Okay. So, if you may have taken say 50 millimolar glycine pH 2.5, right, and you have done a regeneration, and you see, so you run the first cycle, you get your curve, right, and you do a regeneration with your desired regeneration condition. Then you run exactly the same cycle again the second time. If your regeneration cycle is good, then you are supposed to get the same response unit, because you are using the same concentration of analyte. Okay. If you do not, if it goes down, then it is not optimal. But at this point you do not know whether it is, with this one cycle you do not know whether it was harsh or it was mild, right. You just know that it was not optimal, your regeneration was not fine. So, you need to do couple more things actually. So, before that let us look at some of the regeneration buffers that we supply, but again these are very generic buffers, you can have them yourself. The regenerations can be low pH, ranging from pH 3.5 to 1.5. It can be salt, sodium chloride, it can be detergents, ethylene glycol. Okay. So, various things, typically similar things that you use in downstream processing also. So, some guidance to find out whether it was the regeneration, how to find out the ideal condition, and there is again a scouting wizard that you can use to check which is your ideal pH condition, right. So, what you do is a minimum run 5 cycles of your use the same analyte concentration, the same pH, and you run 5 cycles of it. Okay. So, what you are seeing out here is the analyte binding response, and at the same time you need to see your baseline, okay, how your baseline is. If you do not, if your baseline is increasing, that is definitely something is stuck on the surface, right. If your baseline is either stable or going down, that means it is too harsh, right. So, let us look at this situation, whereas this one is 5.5. So, you have run 5 cycles with it is 3, 2.5, 2 and 1.5. Okay. 3, 2.5, 2 and 1.5. So, these are the regenerations that done, and you are looking at an analyte binding here and the baseline response here. If you say at 3, what is happening? The analyte binding is dropping, okay, and your baseline is increasing. It is a clear case of what? It is clear, it is too mild, right, because your baseline is increasing, it is truly, it is definitely, it is very mild, you are not removing all your ligand. As a result, your baseline is increasing, right, and this is dropping because your ligand is not available to bind in the next cycle. Okay. And when you go to 2.5, you see a slight difference. Here there is a slight increase, and then here there is a slight decrease. This is probably slightly not explainable because, you know, in earlier, because it was run just after this. So, some spillover from 3, pH 3 is still there. So, not yet, you do not know the conditions yet, but if you look at pH 2, both are stable, kind of like a actually ideal place where you are keeping both your analyte binding as well as your baseline perfect, right. So, meaning that you are stripping out everything, and after each regeneration you are binding the same amount. But if you look at 1.5, your baseline is stable, but your binding is dropping, right. So, when it is too harsh, not necessarily it is going to strip out your ligand. So, your baseline typically remains stable, but your binding goes down. So, that is your harsh condition. So, this is again a very good way to find out your you know, regeneration condition. I think that is, you know, the end of the talk. So, we talked about some, you know, ideas to, you know, how do you do your immobilization, what are the things that you need to take care before you run your samples, your regeneration conditions. I think it will give you a good start when you start with your Biocore assays. Thank you. If you have any more questions, I am happy to answer. Yes, sir.
This SPR, we run the assay with live cells.
Yeah.
So, in live cells, and we have always analyzed single analyte.
Mm-hmm.
So, each can run more than one analytes with live cells.
Yes, you can do it, but in separate runs, right. So, if
because of in Indian tradition, we have used like herbal medicines.
right.
In that case, one analyte, one drug, one molecule will act on some times. But in case in another way, it give more molecules in combination of all.
Together.
React with more than single molecule.
Yes, absolutely you can do those. So, there are ways that you can do you know, multiple, whether, you know, one binding is affecting the other binding, those kind of studies can definitely be done, absolutely. So, you have to do one after another. So, once you have your ligand, you either mix two things and then see the binding, what is your response unit, and then do the bindings in single analytes and then check what is the binding, if the binding has increased with mixing. These kind of things can also be done.
Choosing between the different methods for which we can measure KD, for example, could you please suggest when one would preferably choose SPR?
So, what other methods do you use for KD? Either the cell based method or the or maybe ELISA you can also use to find out the KD, right. So, you know, finding out the KD is not the crux out here. The KD is the affinity value. There are other techniques to find out the affinity value. And based on the KD or based on the affinity value, if you are actually rating your molecules, you might be limited in the knowledge that you have, because two molecules can have the same KD, but they might have different association and dissociation constants, because KD is actually the ratio between the association and the dissociation constant. So, you would choose SPR when you are actually wanting to look at the detail of how the molecule is really behaving, you know, whether it is a fast associator or a slow dissociator or things like that. So, that is the key here.
The temperature at which it can be done from plus 4 degrees centigrade.
Yes, yes.
So, how do you decide that temperature?
So, you know, typically when you look at the papers that is published, you typically do it at room temperature. So, whenever you report a KD value, you mention the temperature at room temperature 25 degrees, 22 degrees, things like that. Okay. Now, if you are, say for example, you do not see a binding at a temperature, room temperature, you might lower it a little bit, you know, 10 degrees, 12 degrees, 15 degrees, or, you know, to check whether it is still binding or not. In that cases you can actually do it. A lot of people want to find out their KD values at 37, thinking that it is more close to the physiological pH to look at it. In that case also you can choose a different temperature.
And you said that degassing system is inbuilt.
Yeah.
Degassing causes sort of cooling, chilling.
Oh, no, so the degassing actually removes all the suspended bubbles or air which is inside the liquid, and you know, if you don't remove them, they tend to create spikes in the response, you know, and you do not get a very neat.
The process itself causes temperature to drop down. It does.
It does. I am probably a little, but I don't. But it still maintains. Well, I don't think so, because your samples are kept at a particular temperature, right? Your analysis is happening at a particular temperature which you are actually setting. So, I mean, even if you claim that, you know, it changes the temperature, then it is still set at the temperature where you're setting, and you know, you get the at least the binding at that. Yeah.
In terms of association and dissociation, is it usually K off determine the affinity, because K on is diffusion method, and under certain conformational change in binding, then the K on is different?
Well, K on and K off are the property of the interactants. You don't change them typically unless you are changing the temperature. They don't change. Okay, so you can only increase the temperature to increase your association. Okay, the rate of association, you push it a little higher. Okay, but it is the property of the interactants. So you don't change them by anything.
I am sure by now you are familiar with the SPR based experimental workflows. You are also pretty confident about the very basic processes involved in doing these experiments till the data processing and data analysis. You also understand that if you are not very clear about experimental design, and if you are not very clear about data interpretation, you may end up with lot of false positives and artifacts. In label-free biosensors, one of the major concerns have been how to ensure that a binding which is seen, is it coming from two biomolecules, or is it non-specific binding? Is there bulk effect just coming from the buffer itself, or some other artifacts which are present on these gold chips? And that is where your understanding of these experiments and nitty-gritties what Dr. Uma talked to you today becomes very crucial to distinguish, delineate that what is the right binding and the data obtained from that, the sensorgram, versus what could be an artifact which comes from the bulk effect. Some of these concepts will be taken further again in the next lecture, where we are going to have a scientist coming from Tata Memorial Center, ACTRAC, who is going to talk to you about applications of SPR technology in biologically relevant problems. Thank you.